The hidden blueprint, unraveling the secrets encoded in human DNA. Before we begin, don't forget to like this video and subscribe to the channel for more mind-bending explorations into the mysteries of human existence. Now, let's dive into one of the most controversial and fascinating questions science has ever encountered. The discovery that would change everything came not from ancient ruins or archaeological digs, but from a laboratory in Germany on a cold winter morning in 2010. A team of geneticists had just completed the first full genome sequence of a Neanderthal, and what they found would shake the foundations of everything we thought we knew about human evolution. Among the data, among the millions of genetic sequences, there was something that should not have been there. Something that violated the fundamental rules of how evolution was supposed to work. The mixing between modern humans and our ancient cousins had left traces in our DNA. But those traces told a story far more complex than simple interbreeding. It was a story of selection, of precision, of something that looked disturbingly like intention. And it would lead researchers down a path that most of them were terrified to follow. For centuries, we have understood evolution as a slow, grinding process. Random mutations occur, and if they provide an advantage, they spread through a population over thousands of years. This is the mechanism that created every species on Earth, from bacteria to whales to humans. It is patient, it is impersonal, and it is governed by mathematical principles that can be measured and predicted. But when scientists began comparing ancient human DNA with modern genomes, they discovered something that made their equations stop working. The changes were too fast. The precision was too perfect. The coordination across multiple biological systems was too synchronized. It was as if someone had taken the human genome and deliberately edited it, like a programmer modifying lines of code in a vast and incomprehensibly complex computer program. This is the mystery that we are about to explore together, and I warn you now that the answers we find may challenge everything you believe about who you are and where you come from. The human genome contains approximately 3 billion base pairs, the chemical letters that spell out the instructions for building and maintaining a human body. For most of human history, this code remained completely inaccessible, locked away in the cells of living people and preserved only in the fossilized remains of our ancestors. We could study bones and teeth, measure skulls, and analyze tools, but we could never see the actual blueprint that made humans different from every other species on the planet. Then, in the latter decades of the 20th century, technology caught up with ambition. Scientists discovered that DNA, despite being an incredibly fragile molecule, could sometimes survive for tens of thousands of years under the right conditions. Cold, dry environments, like caves in Siberia and the mountains of Europe, preserved genetic material in ways that seemed impossible. And once they learned how to extract and read this ancient DNA, a completely new window into human history opened. The first attempts were crude. The technology was unreliable, and contamination was a constant problem. But by the early 2000s, researchers had successfully sequenced fragments of Neanderthal DNA. By the 2010s, they could read entire genomes from samples that were over 50,000 years old. The precision was remarkable, the data was undeniable, and the implications were staggering. For the first time in human history, we could see the actual genetic changes that had occurred as our species evolved. We could identify which genes had mutated, when those mutations appeared, and how quickly they spread through populations. We could trace migrations, identify interbreeding between different human species, and map the exact moments when humanity underwent its most dramatic transformations. But with this new power came new questions. Questions that made many scientists deeply uncomfortable because the answers did not fit the accepted narrative. The process of reading ancient DNA is a marvel of modern science, and understanding it is crucial to understanding the mystery we are investigating. When archaeologists excavate a bone or tooth from an ancient burial site, they bring it to a laboratory where it is treated with extreme care. The sample is cleaned, sterilized, and ground into a fine powder. Chemicals are applied to extract any remaining DNA, which has been broken into tiny fragments by the passage of time and the natural decay of the molecule. These fragments are then amplified using a technique called polymerase chain reaction, which makes copies of the DNA over and over again until there is enough material to analyze. Sophisticated computer algorithms piece together these fragments like a jigsaw puzzle, comparing them to modern human DNA and filling in gaps where possible. The result is a genetic snapshot of a person who lived and died long before written history, a direct window into the biology of our ancestors. What made this technology revolutionary was not just the ability to read ancient DNA, but the ability to compare it directly to modern genomes. Scientists could now see exactly which genes had changed, when those changes occurred, and how they spread through populations. They could identify the precise moment when humans developed lighter skin in response to reduced sunlight in northern climates. They could trace the evolution of lactose tolerance in populations that adopted dairy farming. They could see how humans adapted to high altitudes, to different diets, to new diseases, and to completely foreign environments. Each adaptation was written in the genome, a record of survival and change that spanned tens of thousands of years. But alongside these expected changes, researchers began to notice patterns that did not fit the standard model of evolution. Changes that should have taken tens of thousands of years appeared in just a few thousand. Genes linked to brain development showed sudden shifts with no clear environmental cause. Entire sections of DNA looked as though they had been swapped out and replaced, not gradually altered through mutation and selection. The data was consistent, reproducible, and deeply puzzling. Consider the evolution of the human brain. Our brains are extraordinarily large compared to our body size, and they are far more complex than those of any other primate. This is the feature that defines us as human, that gives us language, abstract thought, creativity, and consciousness. Scientists have identified specific genes that control brain development, genes that regulate the growth of neural tissue, the formation of connections between neurons, and the development of higher cognitive functions. When researchers examined these genes in ancient DNA samples, they found something shocking. The genes related to brain development had undergone rapid and dramatic changes over a period of just a few thousand years. Multiple genes involved in neural development, language processing, and abstract thinking had all changed simultaneously, as though someone had deliberately rewritten the code that controls human cognition. Under normal evolutionary conditions, this should not have been possible. A single beneficial mutation might spread through a population in 100,000 years if the selective pressure was intense enough. Multiple mutations affecting different genes should take even longer, perhaps 1,000,000 years or more. Yet, the genetic evidence showed these changes occurring in a fraction of that time, with no clear environmental trigger that would explain such intense selective pressure. The immune system tells a similar story. Humans carry genes that determine how the body recognizes and fights disease, a critical factor in survival. Ancient DNA reveals that certain immune genes underwent rapid restructuring, with entire sections being replaced or modified in ways that do not resemble typical mutation patterns. Some of these changes align with known disease outbreaks, suggesting adaptation to new threats, but others have no clear environmental trigger. Populations that were not facing unusual health challenges still underwent dramatic immune system changes. It is as though the immune system was being upgraded, improved, and refined according to some plan or design. Metabolism is another area where the genetic record shows unexplained acceleration. Genes controlling how the body processes food, stores fat, and regulates energy shifted quickly in certain populations. The ability to digest lactose as an adult, for example, emerged in European populations roughly 7,000 years ago. This is an extraordinarily rapid change, happening in just a few hundred generations. While this change is often credited to the adoption of dairy farming, the speed at which it spread remains difficult to explain through natural selection alone. If lactose tolerance provided such a strong survival advantage, why did it not emerge earlier? Why did it not spread faster? And why did it appear almost simultaneously in multiple populations that had adopted dairy farming? What makes these changes even more puzzling is their precision. Evolution works through random mutation, a messy process that produces many failures for every success. Scientists expect to see genetic variation, dead ends, and leftover traces of failed adaptations. But in many cases, the shifts in human DNA look clean, as though specific genes were targeted and altered without the usual trial and error. The missing transitions, the lack of intermediate forms, the simultaneous changes across multiple systems all point to something unusual. Researchers have searched for explanations within conventional evolutionary theory, and they have found some that work for isolated cases. Population bottlenecks, where a small group survives a catastrophe and repopulates, can create the appearance of rapid genetic change. Selective sweeps, where a beneficial mutation spreads rapidly due to intense environmental pressure, can also accelerate evolution. But these mechanisms have limits, and they do not fully account for the patterns seen in human DNA. The changes are too fast, too coordinated, and too widespread. This is where the data becomes uncomfortable, where the explanations start to break down, and where scientists begin to ask questions that they are afraid to ask out loud. The Neanderthal paradox is perhaps the most striking example of this discomfort. Neanderthals were not the brutish cave dwellers of popular imagination. They were sophisticated hunters, skilled toolmakers, and successful survivors who thrived across Europe and Asia for hundreds of thousands of years. Their brains were actually slightly larger than ours, and evidence suggests they had complex social structures, used tools, controlled fire, and possibly had language. When modern humans migrated out of Africa roughly 60,000 years ago, they encountered Neanderthals and interbred with them. The genetic evidence is undeniable, with most people of European and Asian descent carrying between 1 and 4% Neanderthal DNA in their genomes. On the surface, this seems like a straightforward story of two species meeting and mixing. But the details reveal something far stranger, something that suggests the mixing was not random, but carefully controlled. When scientists examined which Neanderthal genes survived in modern humans, they found a pattern that defies random interbreeding. Certain traits were kept, others were systematically removed, and the distribution looks anything but accidental. Neanderthal genes related to skin and hair, useful for adapting to cold northern climates, remain common in European populations. Genes affecting immune function, particularly those helping fight unfamiliar pathogens, also persisted and spread. These make evolutionary sense, beneficial traits that helped modern humans survive in new environments. But other Neanderthal genes disappeared almost entirely, and the speed of their removal raises questions that mainstream science has difficulty answering. Genes affecting brain development and cognition, despite being present in early human-Neanderthal hybrids, were filtered out of the population within a relatively short time. Studies show that regions of the genome linked to neural function are almost completely devoid of Neanderthal ancestry, even though those same regions should have been passed down through normal reproduction. Something actively worked against the survival of Neanderthal cognitive traits in the modern human genome. This is not a small detail. This is a massive pattern that suggests deliberate selection, as though someone was editing the human genome, keeping the traits they wanted and removing the traits they did not. The same selective pattern appears in genes related to fertility and reproduction. Neanderthal variants affecting testicular function and sperm production are significantly underrepresented in modern humans, suggesting they caused reproductive problems in hybrids. This could explain why some Neanderthal DNA was lost as natural selection eliminated traits that reduced fertility. However, the precision and speed of this filtering, combined with the retention of other traits, creates a picture more complex than simple hybrid incompatibility. It looks less like nature taking its course and more like a deliberate editing process, as though someone with knowledge and capability was guiding which genes survived and which were eliminated. What makes this paradox even more intriguing is the geographic variation. Neanderthal DNA is not evenly distributed across modern human populations. People of African descent carry almost none, which makes sense since Neanderthals lived primarily in Europe and Asia. But among non-African populations, the amount and type of Neanderthal ancestry varies in ways that suggest selective retention rather than random inheritance. Certain populations kept more immune-related genes. Others retain different metabolic variants, and the distribution follows patterns that look intentional. Researchers have also found evidence that some Neanderthal genes were reintroduced into populations multiple times, as though specific traits were being selected for and reinserted. This goes beyond normal interbreeding, where genetic mixing happens once and genes either survive or disappear over generations. The pattern suggests active selection, with certain Neanderthal traits being preserved and propagated while others were excluded. The mechanisms for how this could happen naturally remain unclear. Scientists have proposed various explanations, but none of them fully account for the precision and speed of the filtering process. The timeline adds another layer of mystery. The filtering of Neanderthal DNA from modern human genomes occurred relatively quickly, within 10 to 20,000 years after interbreeding began. For evolution, this is an extremely short period, especially for changes affecting so many different genes across multiple systems. Natural selection can remove harmful traits over time, but the coordinated elimination of specific gene variants while preserving others requires a level of selective pressure that scientists struggle to explain. It is as though the human genome was being carefully curated, with someone deciding which traits to keep and which to discard, which genes to amplify and which to suppress. Mainstream science has never been short on explanations for rapid evolutionary change. When confronted with the puzzling patterns in human DNA, researchers turn to established theories, mechanisms that have driven adaptation throughout the history of life on Earth. Environmental pressure, climate shifts, competition for resources, and survival challenges have all shaped species over millions of years. The question is whether these forces can account for the speed and precision of changes seen in the human genome. Climate change is often cited as a major driver of human evolution. The planet has experienced dramatic temperature swings over the past 100,000 years, with ice ages alternating with warmer periods. These shifts forced human populations to adapt or die, creating intense selective pressure. Groups that could tolerate cold, find food in scarce conditions, or migrate to new territories survived, while others did not. Over time, this pressure could favor certain genetic traits, spreading them through populations. The theory makes sense for some changes. Lighter skin in northern populations, for example, can be explained by reduced sunlight and the need to produce vitamin D more efficiently. Cold-adapted traits, like changes in body fat distribution and metabolic rate, align with survival in Ice Age Europe. Genes related to high-altitude adaptation in Tibetan populations clearly respond to environmental conditions. These are textbook examples of natural selection at work, and the genetic evidence supports them. But climate and environment do not explain everything. The changes to brain development genes, the restructuring of immune systems, and the rapid spread of cognitive traits do not correlate neatly with environmental shifts. There is no clear climate event that would drive simultaneous changes across so many different biological systems. More importantly, the timeline does not match. Environmental adaptation works over tens of thousands of years, not the compressed time frame seen in the genetic record. Even dramatic climate change requires time for mutations to arise, spread, and become fixed in a population. The genetic evidence shows changes happening far faster than this process should allow. Survival pressure from disease offers another explanation. When populations encounter new pathogens, those with resistant genes survive and reproduce, passing immunity to the next generation. This mechanism has shaped human evolution significantly, with evidence of past plagues and epidemics written into the genome. The Black Death in medieval Europe left genetic markers in descendants of survivors. Smallpox, malaria, and tuberculosis all left their fingerprints on the human genome. But again, this explains isolated changes, not the coordinated shifts across multiple systems that characterize human genetic history. A disease outbreak might drive selection for immune genes, but it would not simultaneously change brain development genes, metabolism genes, and cognitive genes. The pattern we see in the human genome suggests something far more comprehensive and coordinated than any single environmental pressure could produce. Competition with other human species has also been proposed as a driving force. When modern humans encountered Neanderthals and Denisovans, they competed for the same resources. Groups with cognitive advantages, better tools, or superior social organization would have thrived while others declined. This could theoretically accelerate selection for traits like intelligence, language, and cooperation. However, competition alone does not account for the precision of genetic changes or the selective retention of specific Neanderthal genes, while others were eliminated. If modern humans were simply outcompeting Neanderthals through superior cognitive abilities, why would they retain any Neanderthal genes at all? And why would they retain some genes while carefully filtering out others? The pattern suggests something more deliberate than simple competition. Population bottlenecks, events where a small group survives a catastrophe and repopulates, can create the appearance of rapid genetic change. When a population shrinks dramatically, genetic diversity decreases, and certain traits become more common simply because fewer individuals carry different variants. This phenomenon, known as genetic drift, can shift the genetic makeup of a population quickly. Yet, bottlenecks also reduce overall diversity and create inbreeding problems, neither of which fully explains the patterns in human DNA. The human genome shows not a reduction in diversity, but a carefully directed change in which variants are present and which are absent. This is the opposite of what we would expect from a bottleneck. The most uncomfortable reality is that these explanations work individually, but fail collectively. Each mechanism can account for specific traits or isolated changes, but none explain the broader pattern of rapid, coordinated, and precise alterations across the human genome. Natural selection requires time. Random mutation produces messy results, and environmental pressure does not target specific genes with surgical precision. The data shows something that looks less like gradual adaptation and more like intentional modification. And that is where conventional science reaches its limits, where the explanations stop working, and where researchers begin to feel the ground shifting beneath them. There is a line in modern science that few researchers are willing to cross publicly. Careers are built on peer-reviewed evidence, reproducible experiments, and theories that fit within accepted frameworks, suggesting that human evolution was guided, accelerated, or altered by something outside natural processes is professional suicide in most academic circles. Yet privately, in conversations away from journals and conferences, some scientists admit that the data points toward conclusions that make them deeply uncomfortable. The resistance is not entirely unreasonable. Science has spent centuries fighting off supernatural explanations, insisting that natural phenomena have natural causes. The success of this approach is undeniable, producing medicine, technology, and an understanding of the universe that would have seemed like magic to earlier generations. To abandon naturalistic explanations now, especially for something as fundamental as human origins, feels like a retreat into the darkness of superstition and myth. But the genetic evidence does not care about professional comfort or historical precedent. The possibility that rarely gets discussed openly is that human DNA was deliberately altered, not by gods or magic, but by a process that resembles genetic engineering more than natural selection. The precision of changes, the coordinated shifts across multiple systems, and the compressed timeline all point toward intervention rather than random mutation. The idea is not that evolution stopped, but that it was guided, accelerated, or supplemented by something with intention and capability. This concept is not purely modern speculation. Ancient cultures across the world told stories of humans being shaped, upgraded, or modified by beings from the sky or distant places. Sumerian texts describe the Anunnaki creating workers by altering existing creatures. Hindu scriptures speak of cycles where humanity is refined and recreated. Indigenous traditions from the Americas to Africa contain narratives of ancestors being taught, changed, or improved by external forces. Mainstream archaeology dismisses these as mythology, symbolic explanations for natural processes that ancient people could not understand. But what if those stories preserved a memory of something real? What if the ancient texts and oral traditions were not metaphors, but records of actual events, accounts of human origins that have been passed down through thousands of years? The ethical implications of such a possibility are staggering. If humans were deliberately altered, then questions arise about autonomy, purpose, and identity. Were we modified to serve a function, to fulfill a role in some larger plan? Did the intervention benefit us, or did it come with costs buried in our biology? The idea challenges not just scientific orthodoxy, but fundamental assumptions about what it means to be human. It suggests we are not the product of random chance and survival, but the result of deliberate design. And that raises uncomfortable questions about who we are and why we exist. Are we the creation of some advanced civilization? Are we the product of genetic experimentation? Are we the result of some cosmic plan that we are only now beginning to understand? These questions are not comfortable, and they are not easy to answer. Research into these questions faces significant obstacles. Funding agencies are reluctant to support work that strays too far from conventional models, and scientists who pursue controversial theories risk marginalization. Data that does not fit accepted narratives is often shelved, explained away, or ignored rather than investigated further. Some researchers have noted gaps in the genetic record, sections of DNA that appear incomplete or inaccessible, raising suspicions that not all information is available for study. Whether this is due to natural degradation, technical limitations, or something else remains unclear. There are also institutional pressures to avoid conclusions that might provoke public panic or undermine trust in science. If evidence emerged that human evolution was artificially influenced, the implications for religion, philosophy, and social order would be profound. Governments, academic institutions, and scientific bodies have strong incentives to maintain stability, and controversial findings are sometimes quietly suppressed or reframed to avoid disruption. This is not conspiracy, but caution, a reluctance to open doors that might be impossible to close. The unspoken possibility lingers in the background of genetic research, acknowledged in private but rarely addressed in public. The data keeps accumulating, the patterns keep appearing, and the explanations keep falling short. Mainstream science insists that natural processes will eventually account for everything, given enough time and research. But the timeline keeps tightening, the precision keeps increasing, and the question keeps growing louder. If the human genome shows signs of deliberate alteration, and if conventional evolution cannot fully explain what happened, then what force reshaped humanity? And why does acknowledging that possibility remain so dangerous? The human genome has been fully sequenced. Scientists have mapped every gene, identified thousands of variants, and compared DNA across populations and time periods. The genetic code that makes a human is no longer a mystery in the technical sense. Every base pair, every chromosome, every segment of DNA has been cataloged and stored in databases accessible to researchers worldwide. Yet, despite this completeness, there are parts of the genome that remain fundamentally unexplained, sections where function and origin are still unclear. Roughly 8% of human DNA comes from ancient viral infections, fragments of genetic material inserted by retroviruses millions of years ago. Most of this is considered junk, inactive remnants with no current purpose. But some of these sequences play active roles in development and immunity, their original viral function repurposed for human biology. Scientists do not fully understand how or why certain viral DNA became integrated and functional, while other sections remain dormant. The presence of this foreign genetic material raises questions about how much of the human genome is truly human, and how much was borrowed, inserted, or acquired through processes still not completely understood. There are also long stretches of non-coding DNA, regions that do not produce proteins but appear to regulate gene expression and control biological processes. For years, this was dismissed as evolutionary baggage, leftover sequences with no real function. Recent research has shown that much of it is active and important, influencing everything from brain development to disease resistance. But the mechanisms are complex and not fully mapped. Scientists know these regions matter, but they do not always know what they do or how they came to exist in their current form. It is as though the genome contains instructions that we have not yet learned to read, a language that we are only beginning to decipher. Certain genes in the human genome have no clear evolutionary predecessors. They appear uniquely human, with no obvious relatives in other primates or earlier hominins. Some of these genes are involved in brain function, language, and abstract reasoning, the very traits that define modern humanity. Their sudden appearance in the genetic record, without the expected transitional forms, remains one of the unsolved puzzles of human evolution. Standard explanations involve rapid mutation and positive selection. But the lack of intermediate stages makes the story incomplete. How did these genes arise? Where did they come from? And why do they appear so suddenly in the fossil and genetic record? Then there are the gaps in ancient DNA itself. Not every population has been sampled. Not every time period has been sequenced. And not every individual left recoverable genetic material. The picture scientists have assembled is based on fragments, snapshots from specific locations and eras. There could be entire chapters of human genetic history missing. Events that occurred in regions where DNA does not preserve well, or among populations that left no trace. The conclusions drawn from available data are only as complete as the data itself, and the data is far from comprehensive. We are looking at a puzzle with most of the pieces missing, trying to understand the whole picture from a few scattered fragments. Some researchers have noted anomalies in the structure of certain genes, patterns that do not fit typical mutation processes. These are not errors or contamination, but genuine oddities. Sequences that look as though they were inserted rather than evolved. The scientific community has been cautious about interpreting these findings, preferring to wait for more data rather than draw premature conclusions. But the anomalies persist, and with each new study, the list of unexplained genetic features grows rather than shrinks. It is as though the more we learn about the human genome, the more mysteries we uncover. The more we understand, the less sense it makes according to the conventional model of evolution. The most unsettling conclusion is that the human genome, despite being sequenced and studied extensively, still holds secrets. There are questions it raises, but does not answer, patterns it reveals, but does not explain, and gaps it contains, but does not fill. Evolution provides a framework for understanding human origins, but the framework has holes. Places where the data does not fit neatly, and where alternative explanations become harder to dismiss. If humans were altered once, the evidence would be written in DNA, and if that evidence exists, but remains unexplained, misunderstood, or inaccessible, then the most important question is not what happened in the past, but whether it could happen again. The genome is a record, but it is also a blueprint, and blueprints can be read, copied, and modified. The code that makes us human is no longer locked away in bone and stone. It is open, readable, and vulnerable. Scientists around the world now have the ability to edit human genes with unprecedented precision. CRISPR technology allows researchers to target specific genetic sequences and alter them with remarkable accuracy. What once seemed like science fiction is now routine laboratory procedure. We have the knowledge and the tools to reshape the human genome deliberately and intentionally. We can remove disease-causing genes, enhance cognitive abilities, increase physical strength, and alter virtually any trait we can identify. The question is not whether we can do these things, but whether we should, and whether, in doing so, we might be repeating something that was done to us in the distant past. Consider the implications. If human DNA was deliberately altered by some advanced civilization or external force in ancient times, then we are the product of genetic engineering. We are not the random result of natural selection, but the deliberate creation of some intelligence with the knowledge and capability to reshape life itself. That intelligence might have had goals, intentions, and purposes for humanity. We might have been designed to fulfill specific functions or to serve specific roles. The very concept of human free will and autonomy becomes questionable if we are the product of deliberate design. Are we truly free, or are we following a script written into our DNA by forces we do not understand? Are our choices our own, or are they the inevitable result of genetic programming? These are not comfortable questions, and they are not easy to answer, but there is another possibility, one that is perhaps even more unsettling. If humans were altered once, and if the evidence of that alteration is written in our DNA, then perhaps we are not finished being altered. Perhaps the process is ongoing. Perhaps there are forces, whether human or otherwise, that continue to shape and modify the human genome. Perhaps we are not the end result of some ancient genetic engineering project, but merely a stage in an ongoing process of human transformation. The technology now exists to make such modifications. Scientists can edit genes with precision. We can create designer babies with enhanced traits. We can potentially extend human lifespan, increase intelligence, and eliminate disease. In the near future, such modifications might become common. Wealthy individuals and nations might gain access to genetic enhancement technologies, creating a biological divide between the enhanced and the unenhanced. The wealthy might literally become a different species, separated from the rest of humanity by genetic differences as profound as those between humans and Neanderthals. This is not speculation about a distant future. This is the trajectory we are already on. The technology is advancing rapidly, and the ethical frameworks to govern its use are still being developed. Some countries have banned human genetic modification, while others have embraced it. The global response is fragmented and inconsistent. In this environment of rapid technological change and uncertain governance, it is worth asking whether we are about to repeat the process that created us. Are we about to become the architects of a new form of humanity? Are we about to deliberately modify our own species in ways that will be irreversible? And if so, what responsibility do we have to future generations who will inherit the consequences of our choices? The mystery of human origins, as written in our DNA, is ultimately a mystery about ourselves. It is a question about who we are, where we come from, and what we are capable of becoming. The genetic evidence suggests that our evolution was not purely natural, that something guided, accelerated, or supplemented the normal processes of mutation and selection. Whether that something was an advanced civilization, an external intelligence, or some process we do not yet understand remains unclear. But the evidence is there, written in the code that makes us human, waiting to be fully understood and acknowledged. The implications are profound. If we accept that human DNA shows signs of deliberate alteration, then we must accept that we are not the product of random chance, but of intentional design. We must accept that our species has been shaped by forces beyond our understanding, and that we are only now beginning to develop the knowledge and capability to understand what was done to us. And we must accept that we now have the power to do to ourselves what was done to our ancestors. We have the knowledge to read the genetic code. We have the tools to modify it. And we have the capability to reshape humanity according to our own designs and intentions. This is where the mystery becomes urgent. This is where the questions become not just academic, but deeply personal and practical. What will we do with this power? How will we use the knowledge we have gained? Will we repeat the mistakes of the past, or will we learn from them? Will we modify ourselves in ways that benefit all of humanity, or will we create new divisions and inequalities? Will we enhance ourselves at the cost of losing something essential about what makes us human? These are the questions we must ask ourselves as we stand at the threshold of a new era in human history. The DNA does not provide easy answers. It provides evidence, patterns, and mysteries. It shows us that our evolution was not what we thought it was, that our origins are more complex and more intentional than we have been willing to admit. It shows us that we are the product of modification, whether we like it or not. And it shows us that we now have the power to modify ourselves for better or worse. The choice is ours. But before we make that choice, we must understand what we are choosing. We must understand our own nature, our own origins, and our own potential. We must read the code that makes us human, not just as a scientific exercise, but as a profound exploration of identity and purpose. The mystery hidden in our DNA is ultimately the mystery of ourselves. And the only way to solve it is to look honestly at what the evidence tells us, even when that evidence challenges our most fundamental beliefs about who we are and where we come from. The genetic code is waiting to be read. The patterns are waiting to be understood. And the future is waiting to be written by us in the DNA of generations yet to come. Thank you for watching this exploration into the mysteries of human DNA. If this video has sparked your curiosity and challenged your thinking, please like and subscribe to the channel. Your support helps us continue to investigate the deepest questions about human origins and the future of our species. Don't forget to click on the next video to continue this fascinating journey into the unknown. The answers are out there, written in the code that makes us human. And together, we will uncover them.